And thanks to everybody for the warm welcome. It's been a set of interesting conversations today, and I look forward to more interesting conversations tomorrow. I went back and looked at my title, and it's a little cumbersome. Maybe the question is, how do we get this right? Right? It's the need is real. How are we going to advance the scholarship in biology education? And we have a particular take on it. And as Joe said, I'm going to focus on the CUBES project, but I have to do a little bit of lead up to it. So there's this open question as to whether the kinds of reform that are going on at this point are productive in the long term or sufficient for the nature of the challenges that we're facing, right? The educational landscape is changing. We have all sorts of alternative ways to get certified, to train. And so where's the university's role? Where are other institutions of higher education's role? Maybe this is a better metaphor. We're tending our own little yard very carefully, worried about keeping the grass down. But if you step back a little bit, there's some challenges on the horizon. And so we need to be thinking outside of the fence there, or a little bit larger. And my goal here today is not to tell you how to teach quantitative biology. That just doesn't scale, right? We're thinking about ways to engage faculty, students in using quantitative approaches more broadly and building an infrastructure and systems that will allow that to be a continuous improvement process, because the science is going to continue to change. So changing anything as big and as diverse as undergraduate biology education is much like a complex adaptive system, right? You've got your dependencies and your hierarchies and scaling issues that make all of this extremely complicated, because when you poke it here, you're not sure exactly where the changes are going to occur. And like a complex adaptive system, you have to sort of choose where you're going to work. So the policy, the calls to action are numerous, and I'm going to zing through this because I really want to get to sort of the more interesting things. These are great reading, by the way. If you want to get excited about biology, new biology for the 21st century, it's all interdisciplinary. It's all trying to move into applied problems. This is about the science. And it has some recommendation for education. This one's pretty much straight up education. Bio 2010 came out a while ago now. Obviously the target date was 2010 for the impact, but they had very specific expectations for quantitative skills in undergraduate biology, and that was an interesting project. Howard Hughes and American Medical Association is getting in on this. This has lists of skills for future physicians and for the medical training, and it includes a surprising amount of quantitative skills, very specific suggestions. This is the one that really has gotten the ball rolling in undergraduate biology education, sponsored primarily by the National Science Foundation and carried out by AAAS, a series of conversations on a national level trying to articulate the goals and the next steps for moving away from teaching the old school biology in terms of what we already know and moving into engaging students and building new knowledge. This is advanced placement. Actually, their materials are the model that I use. It was the sort of the best articulated, but it's, these are the 7 science practices, and it's just chock full of quantitative skills, data work. And a lot of this is going to be driven by how we test and assess student success, and so changes in the MCAT, even though they rolled back some of the more quantitative pieces, are significant and beginning to draw attention. And I throw this one in because I'm going to come back to it at the end of the talk, envisioning the data science discipline, the undergraduate perspective. It's not released yet. This is an interim report from the National Academies, but data science is hot, and it's taking over. Everything's going to be talking about data science. So this is an ongoing conversation, right? These are not new challenges. There we go. And to summarize it, I kind of like the way that James Collins does it. He says, look, does what we're doing in the classroom look anything at all like what we do in our labs or in the field? And, you know, we're experiencing explosions, growth of information. The technology's changing all the time. We're having to be pushed out of our discipline to collaborate with others. And this distance between where students learn and what we're doing in school, right? We need the schooling sometimes needs to catch up with that. So this is a kind of a complicated graph. It's not super important, the details, but this broad landscape of undergraduate biology education, right? You've got your departments, and you've got your institutional practices, and you've got your funding for work. I operate in this small, relatively small space. My work happens in terms of thinking about the goals of undergraduate biology education, working with faculty to make incremental progress and build systems that can lead to this scholarly approach so that we're not simply one and done. So I just wanted to position myself a little bit more narrowly than the entire landscape of undergraduate biology education. Okay, so in the next 30 minutes or so, I will take you through kind of a big picture because I wanted to provide some context for what the current project is doing, and then at the end I want to say a few things about where I think this is headed. So the first section will really address this issue of how do we keep the science at the center of our teaching. And to address that, I'll introduce the idea of productive disciplinary engagement, which will provide a framework for thinking about curriculum change and teaching practice. Then we'll move into the Cubes project, where we're really thinking about how to take this sort of individual actions or single project level actions and scale it up and make it community-wide and essentially change the culture, right? Nothing humble about this. We're trying to change the culture of undergraduate biology education. And in order to address that, I'll introduce the concept of scientific gateways for biology education. And then my claim is that we're in the beginning the 4th year of a 5-year grant, and I think we've got something. And so we're now turning to look around and try to understand where's a really hard, important challenge that we can take this machinery and put it to work, right? Try to apply that. And one of those areas involves the notion of data acumen, which I will introduce towards the end. Okay? So that should help us stay organized, and I'll come back to this slide as we move through. And it's, please, feel free to interrupt. You know, sometimes it's acronym heavy. Don't hesitate to ask a question if something comes up. So it is not trivial to design something that reflects disciplinary practice, given that we're dealing with non-disciplinarians, right? We're trying to draw them in, but we have blind spots that develop from our own immersion in our fields. And so I turn back to the philosopher Philip Kitcher for his definition of disciplinary reasoning. And it's really many, many things, right? It's all these aspects of what we do, not just individually, but as a community, right? And sometimes sub-communities that need to be attended to if we're going to draw neophytes in and try to bring them to understanding our epistemology, right? The ways that we build knowledge. And so I really like this framework from Engel and Conant called productive disciplinary engagement, because it provides some guidance. This is their framework for identifying interactions that will support the disciplinary learning. So students have to be invited to problematize the intellectual subject area, right? So it's not simply, here's what we know, right? The textbook is a summary of the conclusions. And then they have to be given the authority to drive some of that discussion. They have to be able to try, without fear of retribution, to make a contribution, right? To build something, to contribute to the community, even though it may be the classroom level community. But it's not a pat on the back because you tried hard, right? You have to introduce this notion of a disciplinary accountability. So we do have expectations. We have measures, or we have standards for deciding whether something is a fruitful answer or not. So introducing those and being explicit about those is important. And then we've got to give them the tools, right? We can't expect them to work with toy data sets and toy tools and actually be able to engage like this. So it's a high standard, but the difference is, right, we're really trying to set them up for doing small versions of the work in the discipline. So my work in this area grows directly out of my experience with the BioQuest Curriculum Consortium, which has been around for 31 years. I've not been there the entire time, but I've had a leadership role since the mid-90s. And this is a loose affiliation of undergraduate biology faculty, mathematicians, computer scientists. We've got some chemists, physicists. And the primary agenda is to address this issue of how do we get students doing appropriate science in the classroom. And generally, we all recognize the value of solving problems, right? And that's traditionally what happens in a classroom, is we'll pose some problems for students and maybe give them some examples, work a couple problems for them, and then expect them to pick up and run with that. But this productive disciplinary engagement model suggests that we need to push out in both directions from that narrow view of disciplinary practice, right? Problems in the discipline don't drop out of thin air. But maybe for some grad students, they land on their desk and they're told what to do. But the idea of understanding what has come before, organizing it, finding a gap, recognizing what's soluble, what would be silly to approach, owning it, right? Committing to an idea. And so this logo here, the 3 Ps, sort of encapsulates this philosophy. It's actually got 5 Ps in it. But it pushes out on both ends of problem solving, trying to get students to engage in the definition of, articulation of problems. And then, okay, you've done some work on that problem, convince me, right? If we're trying to build something, then the practice of using those disciplinary expectations to present your argument is an important component of engaging in the broader disciplinary reasoning. So I'm going to give a couple of small examples from BioQuest and things that I've done in collaboration with some people in that community. Anybody familiar with the RMBL, Rocky Mountain Biological Lab? Yeah, great place. So Billy Barr, you guys met Billy Barr? Yeah, okay. And NOAs. So, you know, it's a treasure trove of long-term data up there. And Ian Billick, the director, had gotten some money to organize electronically the data records from the station. And then we turned around and said, hey, wait a minute. If somebody's organized those data records, let's write another grant and make it accessible to students. Right? And so we did that, and we put together a set of resources with some lessons and activities, and we implemented them with several audiences, studied how they worked, got this publication out of it. But it's, and I teach with this in my introductory biology course, using Billy Barr's 30-year data set of phenology. So this is the winter caretaker at the field station, which is high elevation, southern Rockies, very isolated. And so he has kept a notebook for a very long time, marking first sightings of spring flowers, breaks in hibernation for things like marmots, migrants, like the birds that come in. And it's a very rich data set for students to look at because there's this climatological data that goes along with it. And they can ask sort of their own questions and work with that. So there's one example. This is a little bit dated, but it's still pretty relevant. Yeah, if you ever want to get a lot of downloads, write something that students can use, because yeah, this gets hit all the time. So I'll tell you just a little story with this. Dave Baum was leading it from the University of Wisconsin, and he approached the editor, said, I've got this idea, I'd like to write this little perspectives piece. And we pitched it as an education, right? We are not currently teaching trees to undergraduates, and we really need to do this because biology is built on this fundamental understanding of relationships. And the editors came back and said, no, you have to write it for scientists. And so we were really nervous because, well, we changed the language, and that wasn't too bad. But we had put together tree thinking quizzes. We put together a set of questions, 10 questions that were very fundamental and sort of picked at some misconceptions that many people had about interpreting phylogenies. And then our claim was that if you sort of could master these basic questions, you could take that reasoning and apply it. So we published in the supplement 2 quizzes. The second one took trees out of science publications, and we wrote questions for them. And we were able to post very complicated trees, but people were using the same ideas. It's just more clades, more branches. And yeah, so that's had some impact. There was, we got nascent funding to hold a working group on that, and a lot of research has been published around that. I didn't actually do much of the research, but I teach with these kinds of materials all the time. Again, this was a disciplinary way of thinking. This was trying to get students into the biology and not simply reading the textbook. So we had a fairly large NSF project called Bedrock, which was kind of a clever little acronym, but it also, we were trying to emphasize that homology was the bedrock of bioinformatics. So bioinformatics was going wild a couple decades ago, and people were comparing things that they shouldn't have been comparing. And so we got a project to try to get bioinformatics education, or the use of molecular comparison structure data, into introductory courses, because people were holding it out for advanced courses. And it was well received. We ran a series of workshops at various institutions, but it turns out we mostly attracted people who were looking for help with their research, right? So they were trying to start taking advantage of sequence data, and oh, here's somebody who's non-threatening coming in with this education perspective. And to us, that means we don't want to just talk to researchers, but that means we're in a sweet spot, right? That means we've got something interesting, because there is a drive to get that work understood. We had a project called BIRD, Beagle Investigation Returns with Darwinian Data. And the story behind this one is that, again, just sort of dating it, when we started the project, one of the trying to get some of the actual data, we went to the California Academy of Sciences, went to the research library, got David Lack's notebook, and photocopied all of his measurements of the finches. So he collected, he went to all the museum collections and took new measurements and put them in a spreadsheet. That was a big accomplishment, right? To digitize the data. So it tells you how that's been a little while. And we got the Cornell Lab of Ornithology to send us all of their Galapagos finch songs. They had 13 of them. They had one representative for each species. And now if you go there, they've got hundreds. They've taken all of that work and digitized it. So this resource, right, the idea was it's really hard to teach evolution if you don't have, you know, parts of evolution, if you don't have the data to work with it. And so we were trying to pull together enough data around a central story that students could engage and start to ask questions. And, you know, it's a great story, rich research history, and this was used in the intro sequence at the University of Wisconsin for over 10 years, these data resources, and I'll come back to it a little bit later. So what came out of these and other projects is we articulated this idea of a problem space, the notion that we weren't writing individual labs. We were writing spaces for students to do work. And actually, we were writing spaces for faculty to do work, right? We think faculty play a central role in good curriculum. There are many projects that will try to write things to bypass, you know, teacher-proof the curriculum so that the faculty can't screw it up. We said, we're going to pull together some tools, some data that will allow you to get at some principles, and then the faculty have to guide the students through it, right? They have to be a mentor. They have to help structure the situation so that the students are doing productive disciplinary inquiry. And so this notion of problem spaces grew out of the Bedrock project as we organized data into spaces that teachers could take advantage of. You know, here's another example. There was some interesting work where they were taking blood samples from chimpanzees and gorillas and started to document all the plasmodium that they were finding. And so you had this whole spatial species differential gradient, and we had the actual sequences, so you could look across. And this one actually was published as part of the AP curriculum, though it didn't really sing to the high school teachers. I'm not trying to own BioQuest or these were all collaborative projects, as everything I do is collaborative. But I just do want to mention that BioQuest and a couple of other large national projects that were long-term were recognized relatively recently by an NSF-funded analysis of programs that have had impact. And in fact, they call them communities of transformation based on the sustained push, right? That you create a space where faculty can keep coming back and develop their scholarship. And BioQuest was also acknowledged by the American Society for Cell Biology with the Alberts Award a couple of years ago. Okay, so that's just a taste. This is going to be, like I said, sort of high level and overpass orientation, but a taste for what it might take to create spaces and use spaces for productive disciplinary engagement. This scaling issue is tough. So NSF funds lots and lots of education projects. They have a whole history of quantitative biology and math reform, calculus reform. Unfortunately, those tend to be local. They don't, you know, you have to write in your sustainability plan, and you have to write in how you're going to disseminate, and you have to talk about how you're going to scale. But once you have the money, you just trying to do your best you can. And so this is where we were going with cubes, was to try to take these, and it's not just BioQuest, right? There are lots and lots of very interesting, innovative projects. But there's this, are you familiar with the last mile problem? This comes from sort of shipping logistics or telecommunications. And the idea is that you've got your strong central pipelines, right? You've got your warehouses, or you've got your trunk lines on your communications network. But as you distribute it, right, you've got this tree topology as you go out to the smaller and smaller users, right? A household or an individual store. And then those users might have particular needs, or they might be available on some times and not on others. So it doesn't translate, right? The core central structure of distribution doesn't map to the end user particularly well. And it's, you know, people study that at a variety of levels, and we just thought it really captured some of the challenges that curriculum distribution and reform and uptake was having. And so furthermore, there's this sort of widely held conception that if we just had a big list of good resources, we could change what we do, right? And you know, and I just want to pause here and use something else of Jim Collins's. He defined this challenge as trying to change in the face of excellence, right? We're pretty darn good at what we do. And yet, there's some sense that we need to do something different. And that's really challenging, given how much of specialists we are, and you know, we are good at what we do, but there's this sense that how do we change? Well, if we just had something better. So how many people are familiar with NSDL? Yep, that's what happened. So NSF spent a couple $100 million to put together this national science digital library, vetted high quality resources for teaching K-12, you know, metadata to every degree so you could find what you needed. The original plan included a whole training social network to get people to use it. That part kind of never happened. So they collected a lot of stuff. I was involved in the BEN, the Bioeducation Network out of AAAS. ASM had a library, BioQuest had a library, and then they were all sort of federated into this National Science Digital Library. Right? Not much impact. It's overwhelming. It's just it's sort of like the assumption that if I say it clearly, you'll understand it. Right? It's an information gap model. Right? That's really not what the challenge is. So the 175 million, and now it sits in a server. I don't know if the server's in California, but there's a group in California that sort of keeps it alive. But they pulled the plug on it. People still just Google when they need something, right? They just Google for the activity. So in reflecting on this, and I was part of that assumption myself, the real barrier to getting more quantitative reasoning into the biology curriculum is supporting faculty, right? There's this pinch point where this last mile problem where the faculty need to be convinced it fits within their value system and their constraints and opportunities at their institution. They've got to have confidence, right? We're talking about trying to do biology that, you know, maps to relatively modern science. And so a lot of people didn't have that training as in their graduate program. We all teach much more broadly than we're trained as scientists. And then furthermore, it's not one size fits all, right? That resource actually is a beginning point for you to make it fit in your classroom with your students. You know, the 2-hour class, the intro class, the upper division class. I teach in a computer lab, you know, so I can do it very differently. And so we were committed to this idea that the repository model, right, that finish it and place it there for other people to use model really wasn't going to work. So this is my sort of self-effacing. So I had a funded project that focused on community college faculty, cyber learning, right, all the cool C3, so you had your cloverleaf, and this is a little chipboard print on it. We were going to get these community college teachers who tend to be under-resourced, and you know, but there's so much online you can do, the tools and everything. I was going to put failed, but I thought it was a little harsh. So I went with not particularly successful, right? And I think we gave it a good try. The two takeaways that I took from it was a mistake to try to target and isolate a certain group of faculty, right? So maybe you don't know, more than 50% of students in undergraduate biology courses are at community colleges. It's about 62%, I think, right? So that's where a lot of the undergraduate introductory biology is going on. But still, they were not interested in just talking to community college faculty. And the other thing was that we worked on the social parts, right? We worked on the mentoring and the community, but it wasn't front and center, right? We had the technology in the front and center, and it didn't drive it in the right ways. So there's a little bit the cart before the horse. As a talk, I get to put the order of things that I want. So I'm going to pretend, before I tell you about cubes, that we had this idea of science gateways in mind. It came in as the project was getting started. I'll give you a moment to read it. Think of these as your online scientific community, right? C. elegans people have their place where they go and they put their data and they talk to each other and they share tools. Modelers are great at sharing their tools online. And then some of these gateways are very small and specialized, and some of them are like NCBI, right? This massive national biotechnology, NC. What is it? NCBI, National. Yeah, I'm really embarrassed. I use it all the time. So there is a large project funded by NSF that is trying to support scientific gateways, right? This is what they're taking as the pressure point for getting science done. If we could help these gateways be more effective, then the scientists who use them will be more effective. And we found out about them and approached them fairly early on and said, hey, we think of ourselves as a science gateway, but we're focused on education. And they said, sure, come on in. And we use the Hub Zero platform for our infrastructure, but they support a variety of platforms, and they're connected to all the big supercomputing projects. And so we think of cubes as a gateway for education, in the sense that we provide space for doing the work that educators are trying to do. We provide tools and resources for them, and we provide social support in the sense that we structure programs so that they can be successful. It's a big, complicated grant. It's a 5-year project, multi-institutional. We run faculty development programs. We partner with everyone and anyone, because that's how we are. This represents the infrastructure that we're built on, and metrics is really trying to help faculty document their scholarship, right? So part of the reward system is if you can put it on your CV, or if you can share with your tenure and review committee that your materials have been used so many times. And of course, we have to document what we're doing to see if it's successful. So we call our faculty programs faculty mentoring networks. They're not local, they're national. Here are 4 of the 8 that are going on right now. These are all with partner projects. This is BSA, Botanical Society. This is an ESA sponsored one. Integrate is an earth science education, and of course you're probably familiar with NEON. So they're online groups that are built with faculty needs in mind. We use our platform, long duration, low intensity, meet every week or 2 for an hour for a whole semester. And the idea is that you learn the material, modify the material, use the material in class, and then you get to debrief with your community, and then you get to share it back through our publication system. And so this challenge of the last mile just takes a great deal of energy, but we get a lot of that energy from all of these partners. You know, I thought it was kind of ironic that at 3:30, sort of conflicting with this talk, is a talk from an NSF program officer about broader impacts, right? So we're people's broader impacts. They say, hey, we've got all this stuff, we don't have any users, how do we do that? So we're sort of a matching game on that. This is just a year or two's, it's a little bit dated, but these are, you can see the numbers, they're generally pretty small. The length is the duration, the topics. So we partnered with Dryad, the data repository. We've done some with Railsbeck, the agent-based modeling author, ESA, HHMI, SymBio, if you're familiar with that teaching material. And the takeaway, we've got paper in preparation, that we've got about a 90% completion rate, which includes implementing the materials and publishing your modification back to the community. What I'm most proud about our group is that we're really reaching diverse institutions and faculty. This is not just research universities, right? This is small colleges, it's 2-year schools, not very many high schools. We're really focusing on undergraduate, but people keep coming back and they're able to use them, and we have heterogeneous grouping. Like I said, in the C3 project, we had focused on sort of a monomorphic group, and now we're absolutely heterogeneous. The way that we think about them and evaluate them is informed by education theory, which I'd be happy to talk about, but I'm assuming that most people are not as interested in. The core of it is that we learn by interacting, we learn by doing, we learn by being mentored. So this is just a clip from our website of some publications. So we have built an open education resources publishing system. What that means is that these materials are published with a license, a Creative Commons license, that allows other people to reuse them and republish them as long as attribution is maintained. And so the old model of publication, in our world, was that you finish, clean it up, dot every I before you put it out there. Now, right, we're teachers. Everything's beta. I'm using it this week, but if I teach it again next year, I'm going to have to revisit it potentially. Old model was you publish it, you put it there, it sits there statically. These are now, because of the licensing, open documents, and this, you just click a button and say modify, and you get the text, you get the files, and you can change them for your setting. This is a big one, is that, most of us close our doors and teach, and it's fairly private, and we're not getting feedback, and there's a lot of sort of personal angst around being judged as a teacher. A lot of it's personality sometimes. But we're trying to turn it to scholarship, right? We want people to approach it systematically, read some of the literature, choose a problem, think about a strategy for addressing it, and then use some existing resources and try to move the needle, as they say. So here's just an example. Teaching Issues and Experiments in Ecology. It was a funded project maybe 15 years ago. People wrote data-rich activities. ESA was a co-sponsor of it. These materials sit up on ESA's website in a repository model. So we began running faculty mentoring networks around TIEE materials, and so we've now, for this module, we've published 12 different versions of it. People have modified it for using it in lecture, using it in a combination lecture and lab, or using it just in a lab course itself. And so now, you know, this constellation of materials around this great idea, right, has enriched them. So people would swap out their local data, right? They'd go to their local forestry database. They've swapped out, this was originally written in Excel, everybody's like, oh, give me R, right? You know, this was written as a 3-hour lab. People are stretching it out over 2 weeks and adding additional things, right? But the core idea is built on it, and all of that attribution is maintained, right? That's our metrics part, so that you can tell the impact of your scholarship, which is, if you can't put your finger on the impact, it's very hard to invest. So here's some of that recognition. ESA has helped us immensely. Teresa Murad, the Director of Education there, is all in on this. And so she, in addition to all of these other things, posts prominently on the ESA site. They've labeled them scholars and share their modules. I mentioned that we're working with just a host of partners that, you know, they have a small NSF project that maybe lasts a year or two, and we can give them a turnkey web infrastructure with sort of all the modern communications technologies built in, plus they're embedded in this network of 4,000 or 5,000 users that can be beta testers or targets of their dissemination. And so that's been fairly fruitful, and this is some of the sort of more organizational partners, but it includes professional societies, large sort of institutes, and really big research projects with an educational and outreach component. It's really very, very broad. So that's really where we are. We have this whole back-end computational infrastructure where you can launch an instance just with a click of a mouse and open up a variety of tools that run on supercomputers out of Purdue. Some people use them, but we're really moving into using these much more deeply. So here's me teaching in my class of 300 using NetLogo, an agent-based modeling program. I used to spend, you know, endless amounts of time trying to help students with operating system problems when they download the software and so forth. Now it runs in a virtual machine. They can fire it up very quickly. I was going to show this live, which is always risky, but I won't. We have a tool for opening up what's called a Shiny app. I mean, we have a Shiny server. People familiar with that? So these are ways to put wrappers around our program so that they're sort of button-driven. But behind it, we've also left this markdown so you can have notebooks that are reproducible. So you click a button, your graph gets saved, you can document it, and then that file is saved so the whole research project is reproducible by the next set of students. So we're facilitating students doing research. We are studying the implementation of new curriculum materials, and we've recently started to be used as a research platform. People are finding that a lot's going on. There's good data streams because it's all online, and so they're catching that, and that's picking up. Okay, I'm going to do this pretty quickly. We've written, so let's see, in the last 2 years, I think I wrote, I don't know, 36 letters. So the community sees there's a need. We've struck some chord. People want to write cubes into their projects. So there have been a couple that have been funded recently that I play an advisory role on, or I'm a PI on. And so this is one, right? Here's high throughput data. What are the special challenges of getting students engaged with high throughput data? Here's one that picked up on this notion of the open education resources, and they're trying to do that specifically around data, big data sets. And here's another one that's bioinformatics education. They defined competencies. They're taking existing bioinformatics materials, bringing them back into cubes, incubating them to be more accessible to introductory courses and to reflect their competencies. The collections folks have lots of data, and Ann Monfils, who's down the road at Central Michigan University, is the PI on this. Lots of big organizational collaborators trying to get biodiversity literacy using museum collections and place-based data. So that brings us back to this call for attention to data science in the undergraduate curriculum. Their recommendations out of this interim report is that really we should be using real data, right? We should be thinking about real problems that students can see acting in the world, and we should be using real tools and real methods. And their overarching goal is this notion of data acumen, that it's a judgment and decision, right? You understand enough about the data and the application of that data and the inferences you're making from it to the problem, that you're developing an awareness which could transcend even if you're not going to go into a profession that's highly technical. This would make you a very informed citizen, understanding how some of these decisions are made. Now there's a question, is this data science stuff just old wine in new bottles? And the statisticians, David Donovan's very well known and talks about data science being defined 50 years ago. Really, we sidestep that because we are trying to stay closer to the research. We know that data management issues are coming up, right? Course-based undergraduate research experiences, or what do you call them here? ARCs, right? Do you have students figuring out how to manage that data? Are you giving them experiences with databases? Those skills are going to transfer, and people have begun to study these data literacy skills pretty heavily. So we've partnered with NEON and some others with a pending proposal, big data skills in ecological and environmental sciences. This is a conference proposal focusing on women and underrepresented groups, trying to get their rich materials out into the hands of users, but even more importantly, sort of raise people's awareness of the availability of data. And then we have a pending project that's really, again, focused on broadening the impacts, broadening the participation, and people are pointing to data science, or we would just call it science, as a potential for that, because if done correctly, it can be accessible to people and draw them in. So I just want to end with Jim Collins's notion of, are we doing in the classroom anything that reflects what we do in the lab? And despite all of these complications, these are not going away, right? This is a moving target as the disciplines move. And so we need to adopt the posture within our teaching context of not solving the problem, but keeping up, right? Continuous improvement model, assessing the outcomes, um, becoming part of a scholarly community and contributing to that scholarly community. Huge debt to many, many collaborators, most of whom aren't mentioned here, but really great set of people. And with that, I'm only a couple minutes over. Apologize. If it raised any questions, I'd be happy to take them. Yeah.
Is the United States head and shoulders above all other nations in doing this kind of work in interdisciplinary life sciences, or is there something to be learned from what goes on overseas?
I always think there's something to be learned by looking at what others are doing. I do think that the U.S. education system, which of course is a vast generalization, right, because it really is a complex beast, but I do think there's probably some more risk-taking, right? And so it's pockets. There are pockets of really interesting things going on. I think it's a very exciting time to be in undergraduate biology education. It's changing really rapidly. There were years where, I mean decades, where there were these calls for reform, but very little change. And now, just in the last, I'd say 15 years, there's really a swell of activity coordinated around getting students back into science. And again, I'd point to the undergraduate research experiences in classes, not just going into labs, and moving into the use of research data, even if you don't collect it yourself.
Are you interacting with people overseas in particular nations?
Not a lot. I've got some British contacts. I have colleagues who go to India and Singapore, but those are not ahead of us from what I've seen. Yeah. Yeah. Right. Right, and if you go to the website, you kind of look around and you're like, whoa, there's stuff, but I'm not sure where I fit, or how did, this faculty mentoring network, the application was due a month ago and they're off and running and doing their thing. So the resources are beginning to accumulate, so you can go and treat it like a library, right? What our approach to that has been through our partners, and especially the professional societies. So ESA, SSE a little bit less so, American Society for Microbiology. I mean, we're really supposed to be covering all of biology, but let me tell you, the molecular biologists are not ready for R, right? It's ecology and evolution are way ahead of the game, and that's where we're finding people who are ready to try these things out, right? This is not converting the masses. Right now, this is giving opportunities to people who are motivated and engaged. But we do really want to reach into non-majors and introductory courses. So we rely heavily on our partners to spread the word. And anybody heard of Cubes before I came here? Couple. Okay, good. So it's a real challenge. We're working on it. Yes. Mm-hmm. Right, right. So we were, this is a very political answer, we were very careful to define our goals in terms of faculty use, right? So it wasn't not reported use, but that they actually put it in the classroom and then posted their products, right, their modifications back. It's a whole different ball of wax to go in there and study student learning, even sort of more nuanced notions of culture change. we're working with, I don't have them up here, oh, NICER, National Institute for the Evaluation of, my acronyms are not coming today. It's a great evaluation team, and they are asking some deeper questions. It's going to take a while to figure out how. We're mostly interested in, do faculty persist, right? We think that if they go through one full cycle of scholarship, from sort of getting exposed to some ideas, defining a problem,